We would now like to invite our first speaker, Professor Dame Sue Hill. She is the Chief Scientific Officer, CSO England, and Senior Responsible Officer for Genomics in NHS. Professor Dame Hill will be sharing on the topic titled NHS Genomic Medicine Service from Cohorts to Clinics. Professor Dame Hill, please.
Thank you so much, Senior Minister, senior leaders from IHCC, from PRECISE, from global genomic organisations, esteemed guests. I'm delighted to be with you this morning and to share the journey that we've been on in the NHS in England in conjunction with Genomics England and other research partners. And I'm really going to focus very specifically on moving from cohorts into the clinic. And just to be clear, because sometimes there is confusion, I am from NHS England rather than Genomics England, although I work with them very closely. So in the UK, we have a very rich life sciences ecosystem which supports the delivery of cohort studies and implementation into clinical service. We've had a long history of running cohort studies since the 1930s. We have a number of different cohort studies, as shown on the right-hand side of my slide here, with a genomics element, including UK Biobank, the 100,000 Genomes Project, now Our Future Health that you're going to hear about at this conference, but also Genes and Health and Born in Bradford. Importantly, in the middle here, in 2014, the MRC published how we could maximise the value of UK population cohorts. In that report, they set out 7 recommendations on how population cohorts could actually, one, both be conducted, but also then adopted. What you're going to hear from me in terms of the journey that we've been on all conform to those 7 key recommendations in that 2014 report. I said already that the landscape in the UK makes it possible to really consider from cohorts into clinics. We have a very rich research and innovation ecosystem with 4 of the 10 top global universities, really considerable research funding coordinated by UK Research Institute, the MRC, and the National Institutes for Health Research. The NHS is a single-payer system and can leverage change within the system. It also has longitudinal health records. We have a very vibrant life sciences sector with over 140 different genomics companies with a turnover of in excess of £2 billion. And then we have the political and health system will. You'll see on the right-hand side the recent quote from the new Secretary of State for Health, not long after they came into power. So they really hit the ground running in terms of their commitment. So they're committed to making the UK a life sciences and medical technology powerhouse, and by ensuring that the NHS works hand in hand with life sciences research institutions and medical technology companies. There's been a strategic alignment now for well over 5 years between a vision for life sciences and a 10-year vision for genomics across the UK, bringing all the different sectors together, which builds on years of commitments through different white and green papers. But I think this statement is really important in terms of the level of commitment that we're going to see from this new government as we move forward. In 2022, we published in the NHS our very first genomics strategy that was focused on accelerating genomic medicine into the NHS, with a vision of genomics being and supporting, predicting, preventing, and diagnosing disease, but then targeting treatments, all accessible as part of routine care. You'll see that there are 4 priorities, one of which is embedding genomics in the NHS through a world-leading innovative service model. Secondly, delivering innovative and equitable genomic testing, and linking that testing not only for earlier diagnosis or prediction and prevention, but with treatments and access to clinical trials. The third priority is enabling genomics to be part of the data and digital infrastructure and available for research, but also for ongoing service development. And finally, to continue to align routine care in the NHS with evolving the contribution to cutting-edge research and innovation. You can see in 2019, just after the completion of the 100,000 genomes, in the NHS long-term plan, there was a commitment to introduce whole genome sequencing as part of routine care. And when we launched the strategy, our chief exec really gave us the backing, the chief exec of the NHS, to take forward the strategy, and recognised it was one of the most exciting bets for the NHS as we move forward. So the 100,000 Genomes project really built on the long-standing genetics profile that it had had in the NHS since its formation in 1948. And it created a national infrastructure and asset from a high-profile cohort study. And that goes back to one of the key recommendations from the MRC report. It was launched in just after the London Olympics by the then Prime Minister. And you'll be aware that we've had many Prime Ministers since then. And we've got a new one from a different party now. But he announced the project. He was very much driven by having a child with a rare disease who sadly died. But they established, as part of this commitment, Genomics England, which is a company owned by the Department of Health and Social Care. And they were set up to deliver the project without some of the difficulties associated with being very close to government. It had 3 main aims: to build the evidence for whole genome sequencing as a diagnostic test, to align genomic research and discovery with routine clinical practice, and to have the conversation with participants about sharing their de-identified data for research and development, and to have access to their longitudinal medical records. This is the overall schema for the 100,000 Genomes project. NHS England invested heavily in the infrastructure that you see in blue, because it recognised that as part of establishing that infrastructure, it would establish the pathways and the infrastructure for adopting whole genome sequencing as we move forward into a clinical service. And you can see the NHS consented patients, collected samples, and also provided a metadata set at the very beginning. After, on the right-hand side, which indicates the Genomics England elements, after their samples had been sequenced by Illumina, run through the semi-automated bioinformatic pipeline at Genomics England, those results came back into the NHS for validation and reporting in NHS genetics labs before they went into newly formed genomic MDTs and then returned to participants. You can see on the right-hand side that Genomics England also established a reading library where industry users and also other scientific users could access that de-identified genomic and clinical data. Over nearly 90,000 participants were recruited, over 110,000 genome sequenced, the bulk in rare disease, so index cases and family members, and 37,000 cancer patients. You can see that overall in rare disease we had a diagnostic yield of 25% up to 60% for some clinical indications. 14% were found in regions of the genome that would be missed by other methods. 50% of cancer patients had actionable results or were eligible for clinical trials. Over 1,000 new genomic findings were identified. As a follow-on from the study, additional look-for findings such as inherited cancer, predisposition syndromes, or familial hypercholesterolemia genomic variants were found in just under 1% of those participants. And in a cohort of the rare disease participants, potentially actionable pharmacogenomics variants were found in 80%. And in a just really recent publication, in the cancer cohort, potential adverse drug reactions have been found, again in a similar percentage to commonly used chemotherapy agents. The project covered a broad range of rare diseases, and I wanted to make this point because this is important as we think about this as a national asset and an asset for ongoing diagnostic discovery or reanalysis. The largest number were in neurology and neurodevelopmental disorders, but also there are a significant number in hematological and immunological disorders or skeletal disorders. Similarly, the project included a broad range of different cancers. What the project did was establish the National Genomic Research Library that is held by Genomics England. And within the research environment, you can see the research environment that's accessed by researchers, and those researchers can be from industry, from academia, from clinical services. That includes genomes, primary clinical data, secondary clinical data, including linking to registry data, such as our cancer data registry, and also NHS reporting outcomes. Currently, over nearly 2,000 researchers have access to this data, so they've been authorized for access. There's over 7 million germline variants that have been available and are within our clinical variant arc. And there's, to date, over 130 different publications that have been used using this National Genomic Research Library data. You can see there's a very clear access gateway here before anyone is allowed in to the research library. The project, importantly, demonstrated the cost of the diagnostic odyssey in terms of the time it took for some individuals to actually obtain a diagnosis, the number of episodes of hospital care and the cost associated with that. So here, just nearly 183,000 episodes at a cost of over 87 million for the participants. And then in unaffected participants, an equally high cost in terms of those individuals. And this really demonstrates, I think, one of the challenges we all have is ensuring that we can reduce that diagnostic odyssey, we can reduce the circle, which is what this infographic is actually showing, this winding pathways that people in our health systems experience. And we can also reduce the cost if we get this right by using the right test at the right time for the right patient. In this instance, whole genome sequencing. What the project has been doing is supporting ongoing diagnostic discovery with feedback to participants and patients in a model that's been developed jointly between NHS England, NHS Genomic Laboratory Hubs and Genomics England, with joint agreement on any variants that are actually fed back into the NHS. And so now that National Genomics Research Library includes both the project genomes, and as you're going to hear from me, also the genomes from the NHS Genomic Medicine Service. But you can see over 3,500 putative new diagnoses have been returned to the NHS, and 85% of those findings have been likely pathogenic or pathogenic. So it just demonstrates the value of the data set. And this was I did a recent article with the Sunday Times, and this patient, Ben, was one of those patients that benefited by us being able to go back and reanalyze his genome when new evidence was made available. He had a negative finding from the 100,000 Genomes Project, but 5 years later, now has been diagnosed as having Marlin syndrome, affecting about 350 people worldwide. And the headline was actually the quote from his parents. So I want to go on now to talk about the Genomic Medicine Service, so building on that project into the clinic. So we established the Genomic Medicine Service in 2018, and it works across the care continuum from primary to secondary and tertiary care. So with about 8,000 general practices, over 220 hospital organisations. It was established to complete what was left of the 100,000 Genomes Project, including those additional findings return, to retain and build public and patient trust, to strengthen the partnerships across the NHS, across all those multiple providers, in 7 geographical areas, as you can show depicted on this slide, ranging from covering 10.1 million people in NHS Northeastern Yorkshire Genomic Medicine Service to 4.5 in the Southwest Genomic Medicine Service. To really use genomics to improve not only health and care, but productivity, quality, and efficiency. To support the multi-professional workforce to use genomics safely and effectively, and to maximise opportunities to facilitate and participate in research and innovation. And we established, because we've got national funding, national leverage, we established 7 genomic laboratory hubs, so we consolidated our previous genetic laboratory infrastructure. In 2021, we wrapped those laboratories around with 7 genomic medicine service alliances to support the embedding into NHS clinical pathways. And we aligned this infrastructure with our 17 clinical genetic services. These are the key organising principles that go back and align with those 7 MRC recommendations that I mentioned at the very beginning. Our focus is on providing equitable genomic technology-driven care for the 55 million people in the population that the NHS in England serves. We have a national genomics unit that I oversee that provides national coordination for this National Genomic Medicine Service with oversight and resources. All the resources are together nationally, which gives you great leverage for change. We have a single funded National Genomic Test Directory that you'll hear from me in a minute, that we can update annually and is within contracts with our 7 genomic laboratory hubs annually. We have funded clinical and scientific leadership You're going to hear from me about the strong patient and public involvement and how we establish national standards and protocols. We're moving to standardized pricing models with a price versus activity model. And we enable rapid access to precision medicines and clinical trials and are aligning with the rest of the NHS data and digital infrastructure. And as part of our research, innovation and transformation agenda, we've established the NHS Genomic Medicine Service Research Collaborative. You can see on the right-hand side what we're trying to do from targeted disease prevention into a really targeted therapy, early disease detection, accelerated diagnosis. But at the bottom here, both improve outcomes for patients, but improve elements for the system in terms of value for money, in terms of productivity, in terms of efficiency. We took forward the partnership between the NHS and Genomics England in 3 areas: in a strategic partner as leaders in the ecosystem of genomics in the UK and often speak as leaders, in a contractual arrangement because Genomics England deliver a range of services for NHS England to support the whole genome sequence service, and in research. And I'm going to come back and show you about the generation study, but we're working on Cancer 2.0, and the photograph just shows one of our genomic laboratory hubs in northeastern Yorkshire, actually working on the long-read developments with the Promethean from Oxford Nanopore in hematological malignancy. We're working also with them on the diversity of data, and this links with the work that I'm leading on blood group genotyping in sickle cell, thalassemia, and rare anemias to really enrich the data set that we have to reflect the diverse population in England. So that comprehensive offer for our citizens is outlined in the National Genomic Test Directory. These, as I said, are nationally commissioned and paid for tests that are within contracts with our 7 genomic laboratory hubs, updated annually. We delivered over 800,000 tests in 23-24. And it wasn't until 2018 that cancer testing, somatic cancer testing, came under the remit of the Genomic Medicine Service. The test directory covers over 7,000 rare and inherited diseases with a genetic cause and over 200 cancer clinical indications, plus pharmacogenomic testing. To date, as part of the cancer offer, where our commitment is to offer more next-generation sequencing panels, we've delivered over 300,000 large panels, pan-cancer panels, either for solid tumours or for hematological malignancy. You can see that the offer is in target testing, and some of that target testing is, for example, for diseases such as Huntington's, but also in that targeted testing, this is inclusive of microarrays. We have undertake quite extensive panel testing, and also in that bucket is all our work in circulating tumour DNA biomarkers. We use exome sequencing for core and specialist rare and inherited disease, and are working on that now for cancer from fresh frozen and paraffin-embedded tissue as part of our commitment to messenger RNA vaccines. And then on the right-hand side, whole genome sequencing is part of the offer, and it accounts for about 10% of our testing. The test directory through our horizon scanning and evidence developments will also expand, and our cancer panels, for example, include RNA targets, but the horizon scanning will mean that we will start to include more elements of the functional genomic pathway, the transcriptome, proteome, etc., as we move forward. There are a number of mechanisms to update this text directory that are reviewed by test evaluation working groups, including by a direct application from clinicians or researchers, or if it's required by a policy or a NICE health technology appraisal, or if it's by an internal review by our lab and scientific team. Since 2018, there have been 350 updates to the test directory. And separately, from the work we do on panels, there's been over 2,500 changes from the panels that are applied by Genomics England in the WGS analysis, or from other panels that are used in next-generation sequencing. These are the changes in cancer, and you can see the biggest one is associated with either the number of tests with gene target changes or on new tests that we've introduced as non-whole genome sequencing, particularly moving from lots of targeted tests into next-generation sequencing panels. And on the right-hand side, you can see 131 types of changes through this process at the top in rare disease, including new disease, but the removal of some tests as we change the test up to whole genome sequencing. As I've said already, NHS England and Genomics England work closely together to deliver the whole genome sequencing. The blue relates to the contribution of what the NHS does. The sequence is provided by Illumina at their site in Hinxton in Cambridge. Genomics England undertake the variant calling and also the automated interpretation before it goes back into the NHS again for validation, further testing, and back to the multidisciplinary team. This can take actually a long time. I'll come back to that in a minute. Interestingly, 92% of patients in the routine clinical service have provided their consent for their data to enter into the National Genomic Research Library. And currently, there are 133,000 whole genomes from both the 100,000 Genomes Project and the NHS GMS in that library. We just surpassed in the routine service over 100,000 whole genome equivalents being sequenced. And we started the service in COVID in November 20, and it was a very slow start because of COVID. And now we will be accelerating the uptake. The diagnostic yield remains at about 26%, suggesting that we'll need to use other whole genome sequencing approaches, such as long read. But in some diseases, like cystic kidney disease, the diagnostic yield is around 60%. You'll see along the bottom there, we are establishing pathways as we move forward to provide whole genome sequencing via the NHS genomic laboratory hubs and a mechanism to bring that non-whole genome sequence data into the NGRL. We have introduced other cutting-edge services, such as our rapid genome sequencing service for acutely unwell children that's been built from research cohorts, led from the unit in Exeter. It started with a research exome, then moved into an NHS gene-agnostic exome, and then in 2022, through various developments of the pipeline, we introduced our rapid genome sequencing service into the NHS. And it's now being developed in terms of a multi-omic approach as part of our network of excellence. This is the service. There's about 3,000 patients that access this each year. They analyze over 22,000 genes and look for over 7,000 rare genetic conditions in this single test. So you can see it's about 1,200 children a year, but we're up to 3,000 that's been through this. And it's a test that really changes both the amount of time that these acutely unwell children spend in intensive care units, but also in the case of Rubin, really was life-changing in terms of the intervention. Sometimes long-read sequencing is also needed to really understand the inheritance patterns, and this was in a child with severe epilepsy, and sometimes looking at protein conformations and modelling. And this has been really important when very difficult decisions have needed to be made about whether there's any intervention with these young babies and whether they would essentially, whether it's a fatal mutation that is present in that particular child. And therefore, the outcome is a more palliative and hospice-based care. We've also taken forward other evidence from other cohorts, including the prenatal assessment of genomes and exomes, the PAGE study. And what this provided was the criteria that should be included in a future service within the NHS. So it demonstrated both the evidence for pre-screening by clinical genetics and defined that eligibility criteria that could increase the diagnostic yield, such that in October 20, we translated this into a nationally commissioned rapid fetal exome sequencing service in England. And you can see that the diagnostic yield is around 30%, the mean turnaround time around 14 days, and it's impacted on the management of 56% of cases. We'll be moving this service into a whole genome sequencing service because of the potential of in utero therapies that are on the horizon, and therefore the potential to influence much earlier in a lifestyle. These are some of the key elements of the service that support our overarching aims, which includes driving transformation through clinical and scientific leadership, working with the system, a system that has multidisciplinary leadership, including clinical, scientific, nursing, midwifery, and pharmacy, and where we've funded innovation directors and research coordination. A clinical leadership model with all the key professional organisations. Our ability to support clinicians and transform services in areas of unmet need, such as Lynch syndrome, sudden cardiac death, epilepsy, FH, renal disease, and monogenic inflammatory bowel disease and diabetes. We have a national genomics education program that's developed significant education and training resources in terms of just-in-time learning, such as clinical pathway initiatives, step-by-step learning for certain genomic pathways, and also reactive learnings and also data sheets. We have a cutting-edge consent model where patients give a clear and informed consent about their choice and also their participation in the NGRL. And this is being developed also for non-whole genome sequence data. Very active public and patient involvement with members involved in all the governance groups, in all that infrastructure I've outlined. And we've established an ethics, equity, and legal advisory group. We have standardized or validated sample collection tools and platforms, including an end-to-end external quality assurance scheme with Gen UK, which is part of UK NEQAS. That means that everything is externally quality assured, right to the stage of variant interpretation, and then also common processes to drive standardization across the system, including in interpretation and reporting and other operational policies. We use data to drive equity of access in terms of monitoring turnaround times, understanding the populations we're testing, the geographies with the greatest needs, and identifying population segments that access a greater proportion of genomic tests. There are a number of challenges that associated with this, but last year we published for the first time our test activity. We also are progressing our genomics data and digital infrastructure, and from something that now that there's a system for whole genome sequencing that was derived from the 100,000, but has limited interoperability with the service in the NHS. There are a number of different elements to being able to scale, and for non-whole genome sequence, that data is siloed within those 7 different genomic laboratory areas. We're moving into a unified genomic record that will link with all the different other data sets for an individual patient, and where that unified record can be updated on an ongoing basis, which will be standards driven, adopting all the national and international standards, but also have structured data sets at a national level. We'll be going into the alpha phase of this next year. We're supporting the use of precision medicines, and I co-chair a genomically informed medicines optimization board that links with the overall NHS medicines optimization approach. And this enables access to new and existing medicines informed by genomics, including gene therapies and advanced therapeutic medicinal products. And this infant diagnosed with metachromic leukodystrophy, diagnosed from the service in Exeter, the rapid acutely unwell child service, was the first NHS patient to receive the life-saving gene therapy Lameldi. We facilitate access to clinical trials when access is genomically stratified, and some of that is associated with our new partnerships in messenger RNA vaccines in cancer. But later this month, we'll be introducing cancer clinical trial targets as standard of care on our NGS panels, starting with 5, and by the end of March, there will be 50 clinical trial targets reported as part of standard of care. We're supporting the safe use of medicines through the use of pharmacogenomics. This is just an example from DPYD, from the use of fluoropyrimidines in the cancer pathways, and really integrating genomics into existing treatment pathways. And that includes working with NICE and with our regulator, MHRA, and this example was just from familial hypercholesteremia. The NHS is at the heart of the NHS innovation ecosystem, and we've just introduced and funded NHS Genomic Networks of Excellence in 8 areas. They're designed to bring together the NHS with academia, the third sector, i.e. patient groups, and also with industry, to leverage funding. And these 8 networks have leveraged over 50 million of research funding. They're designed to gather the evidence to speed adoption of new technologies within a 2-year window, and they're strategically aligned. One of those is actually in respiratory metagenomics, and this is being used as a pathogen biosecurity and biosurveillance system, because it's focused on intensive care and using Oxford Nanopore technology to rapidly sequence those patients with severe infectious diseases. On the right-hand side, this is an example of our rare and inherited disease, working with different partners, but also partnering with LifeArc in the mitochondrial disease and with the MRC rare disease nodes. As we move forward, the further cohort research with Genomics England will include in the generation study, so newborn sequencing for 100,000 newborn babies. There will be within this study 207 different conditions looked for associated with 490 gene condition pairs. It's all been underpinned by clinical discussion and NHS England governance, because that all the recruitment happens in the NHS. Although Genomics England will return a sequence result to a parent, all the validation and confirmatory testing will happen in the NHS, funded by the NHS, and all the ongoing care. And we've established 207 new clinical pathways, including defining confirmatory diagnostic testing, and that might not be genomic, other biochemical and other testing, but also the treatments. And we've actually got that and had those treatments and interventions approved by NICE and MHRA. And we're now live, and we're at over 500 babies that have been recruited. So just to finish, looking towards the future, we're looking to adopt and accelerate our technological, clinical, and scientific developments. We're going to industrialize genomic testing at scale and make genomics commonplace in the NHS. We're going to have a new cancer offer that's across the whole cancer pathway. We're going to have a new rare and inherited disease offer that's multimodal in terms of the types of technologies that will be applied, and introduce a new rare disease launchpad for new therapies. We're going to integrate genomics into NHS data through the Unified Genomic Record, but also making genomic data available to all patients. We're going to deliver pharmacogenomic testing at scale, both in a reactive for areas such as supporting Alzheimer's therapies, in other cardiovascular type of medicines. But then in a proactive way with the work that we're doing to define what would help primary care. And we're going to continue to align our NHS Genomic Medicine Service with precision medicines and also with clinical trials. Then finally, we're going to focus on improved population health to provide more rapid prediction, increased prevention, early diagnosis and monitoring. And some of this will be the evidence and assimilating the evidence for the newborn screening programme, but also from studies like Our Future Health that's looking at population-based integrated polygenic risk scores. All of this relies, as we move forward, on establishing successful partners, being part of a global learning health system. This includes in the UK working with Our Future Health, UK Biobank, HRD UK and Genomics England, but also recognising to do this we need to develop the workforce that's going to receive this. And this is just a photograph from the first meeting of the Global Genomics Network and of in education and training, and many people are in the room today. And this is just my final slide. Our genomics ecosystem vision in the UK is a world where everyone benefits from genomics healthcare. Our NHS genomics mission is to be world leading in implementing genomic medicine to enable patients to access the most effective care and treatments. Thank you very much.
Thank you very much, Professor Damehill, for your insightful presentation. We all benefited from your generous sharing of NHS genomic journey. Thank you, and we invite you back to your seat. Well, distinguished guests, ladies and gentlemen, thank you for joining us for the morning's conference opening. To all participants of the Asian Biobank discussion, please be reminded that this session